Thank you very much. So before I start, I would like to take the opportunity to thank Sakea and also Luisa for putting together such an amazing program of this year's Organic Photonics and Electronics conference. Also would like to thank Ruth Shinar for putting together a very nice also session within the transistor and bioelectronics session. So I will talk about electronic plants and artificial nervous system. Before I do that, I also would like to mention a little bit about the groups, the two groups I'm working with. This is the Laboratory of Organic Electronics at Linköping University, and we are divided into 8 different groups. I also work very closely with a CREO Institute, which is an institute that further develops our science and technology and makes it also producible and also commercialize it into various kind of applications. So before I go into my own results, I would like to discuss a little bit about materials and signals in different kind of processing systems. Let's start with electronics. Everyone knows that we have the diamond, crystal, silicon wafers. The electrons are the signal carriers, where in mammalian systems, typically these 3 components is the triglycerols, is the phospholipids, and the proteins build up the most part of the structure in animals and also in humans. And typical signals within this system to regulate all the physiology, all the growth, and all the processes in animals are represented by typically cations, neurotransmitters, proteins, and also a vast array of other different signaling entities. In plants, you might think plants are very similar to humans, but in fact they differ quite a lot. Even though they come from the nature kingdom, they are actually constructed from slightly different materials. They are typically based on sugar rings, therefore polysaccharides are one of the main components. So cellulose is one of the main components. You also have lignin and other ring-based macromolecules that build up plants. And therefore, most of the hormones that regulates growth, flowering, and all the physiology in plants are then typically ring-based signals. So in one effort, we are trying to bridge the signaling gap between electronics and mammalians, and in another effort, we are trying to bridge the signaling gap between plants and electronics. Before I continue, I would like to also spread the news about there has been tremendous achievements during the last 2 decades in bioelectronics, where different kind of transistors, OLEDs, electrodes, and so on has been used to convert signals from and to electronics and in between electronics and cell system, tissues, and so on. So the first effort trying to develop artificial nervous system, we call this Biocom lab. So if you take a look at the nervous system in, let's say, humans, signals are distributed and shuffled around primarily in dendritic systems. They are chemical circuits, and basically we are using, this system is using AM amplitude and frequency modulation, and in order to achieve demultiplexing and addressing of signals in the nervous system. When signals are traveled along the nerves, along the dendrites, typically this is achieved or entirely achieved by the depolarization and polarization of sodium and potassium, which happens in between the myelinated shielding coatings around the nerves. And finally, at the end of a nerve, the signal is processed. So the depolarization of potassium and sodium is converted into the release of a neurotransmitter across the synaptic cleft. And then this neurotransmitter, small size molecule, is received by proteins, which then generate a new action potential. So that is sort of the computing element in our body. So we are trying to put together different technologies in order to make a parallel nervous system to the one we have in our body. And the main reason for doing this is that we are trying to target different neurological diseases like Parkinson, epilepsy, chronic pain, and so on. So we are introducing a body network, body area network, and we use capacitive coupling in between components running at 13.56 megahertz. We are adding on printed electronics in the form of skin patches and also electronics on medical packages. We have then an organic electronic ion pump that can distribute and dispense neurotransmitters inside the body. We use electrochemical transistors for sensing some of the signals in vitro and also in vivo, and we are running this also in chemical circuits. So let's start with the printer electronics technology. We are aiming for skin patches and also electronics on packages. Many of these technologies has been further developed by a Creo Institute, and also we have different kind of labels where we can do sensing and so on. So this platform is basically composed of 2 silicon chips, one for communication and one for sensing, and then we hook on different kind of printed electronic component depending on the specific application. Most important is that we can add on different kind of sensors, basically all kind of printed sensors that generate a change in current, resistivity, voltage, or capacitance. So we are producing these labels in a recently established facility where we have different kind of tools, not lined up though, but different parallel tools with a total capacity of 5,000 to 10,000 electronic sensor labels per hour. That's what we can produce in this. And this is one example where we have a glucose sensor added on to such a label. And we have done a pre-calibration of this sensor. So this is the calibration curve with a very, very high linearity. And in this case, we add then a sample, and apparently we read something like 300 or 350 nanoamp, which then results in a reading of 3.5 millimolar glucose concentration. And of course, we can add on basically any kind of printable sensor. So if you have any ideas, please come across later on, and we can see if we can actually add this onto our label platform. Another version of this label is this. It's basically a temperature monitor, and we have been collaborating for a couple of years with Médecins Sans Frontières, with our research department, and we then developed this label which goes on to vaccine packages that are distributed and to doctors in rural areas. Yet another version is, of course, this skin patch, which is our next target. This is just a mock-up to illustrate how this could look like. So the next technology is then the organic electronic ion pump. This device converts an electronic addressing signal into the delivery of neurotransmitters, and the goal is to bring this in close proximity to systems where you have some kind of neurological disorder. This device works like this. We have 2 electrodes composed of P.PSS that can make charge compensation and ion exchange. We have an ion-selective membrane where we primarily transport ions along the film. And we have a source and a target electrolyte. So this basically should represent the organ or the system where we want to pump the neurotransmitter that is stored in the source. We add on a voltage, and due to the selectivity in this membrane, only cations can be transported. So if we have a cationic neurotransmitter here, then primarily this one will be transported to the receiving reservoir. And ideally, the number of electrons equals the number of ions. And in many of these devices, we have close to 100% in, let's say, quantum efficiency in these devices. In order to design this and figure out what kind of voltage that is needed, we have to consider both the Donnan potential that is generated because we have fixed charges and we have the electrolyte out here. There will be an exchange of ions of different sign charge, and therefore we all need to consider the Donnan potential. We also have the junction potential, which is basically taking care of the difference in transport numbers of anions and cations that can go through the system, and which also will generate a potential across. And then we apply a voltage in between these 2 electrodes, and then electrophoretically we force the neurotransmitter to transport through. So that's how this device is operated. So basically we have an electronic signal sent in via charge compensation, and electrophoresis, this one is delivered. So further on I will talk about devices primarily that has been shaped into a pen-shaped device structure, so we can put this into close proximity to the organ that we would like to trigger or stimulate. So in one example, we took 4 outlet ion pumps, electronic ion pumps, and put it in proximity to brain slices that is representing epileptic seizures. This is a model system that has been derived at the University of Marseille by Christophe Bernard's team. And we pump, in this case, the neurotransmitter GABA down to this region. And these molecules are then launched, and it's an antagonist for this epileptic seizure. So the idea is then to try to suppress the seizure. And what we find out is that after a few seconds or close to a minute, we see that the seizure is entirely suppressed by this. And if we do the same recording in areas far away from the delivery outlet, we see no impact. So it just keeps expressing epileptic seizures. So I will come back to the speed. Of course, an epileptic seizure is something you would like to suppress or terminate instantly, at least within a few milliseconds or so. I will come back to that later on. In another experiment, we took the same device, reshaped it into a form so we could put it into the spine of rats, another animal model system. And the idea is to have the outlets lined up. So we target the 3rd to 6th dorsal horn within the spine. These are the locations where the pain sensation is generated in our body. So the idea is to have a neurotransmitter, in this case also GABA, delivered at these different points and see if we can actually impact the pain threshold. And the target patients here are patients that are suffering from chronic pain that cannot be treated with pharmaceuticals. So basically this is a chemical resistive circuit, and we try to design all the resistors so we have the same amount of neurotransmitter delivered at the different outlets. So if you look at the response, this is the pain sensation level of a healthy rat, and this is the hypersensitized rat. And if we apply more and more charge to the system, we will see that after some time we have a clear suppression of the pain threshold, not really coming down to healthy rats, but this is a tremendous achievement in terms of most of the other pharmaceuticals have no impact, and around 40% of patients suffering from chronic pain cannot be treated whatsoever. So many of the signaling events and signal processing in biology rely on the fact that a neurotransmitter is released and recorded almost at the same spot, or at least within a distance of a few nanometers. So there has been a great challenge in neuronal signaling, or trying to artificially contact neuronal signaling, is to have both the delivery and sensing in the same electrode. How can you achieve that? So together with the Georges Maliara's team at EMC in France, we basically composited their sensing technique with our ion pump. And this very same electrode which is performing sensing is also the outlet point for our neurotransmitter that we are pumping. So in this experiment, basically we took a mouse hippocampal slice, and the idea is then to pump out GABA, the neurotransmitter, so it impacts the GABA receptor, perform a depolarization. This then is an antagonist to suppress the release of potassium ions. And this change in the release of potassium should then be possible to detect with the sensor. So emission, detection, emission, detection. So basically this is a closed loop of signaling communication across the technology, biology interface. So you can say this is a presynaptic terminal, postsynaptic, presynaptic, postsynaptic, and it can go round and round like this. And we can see that when we turn the pump on, we immediately kill or terminate the emission of potassium, and then we put the increasing agent, and then we start to see signaling again, and then we turn the pump on, and we once again terminate the signaling. I mentioned the speed. Of course, it's attempting to have a pump that can generate or stimulate with neurotransmitters so fast that you can actually terminate the epileptic seizure before it actually kicks in. In order to do that, we need to deliver neurotransmitters at the speed of around 100 milliseconds. That's the time lapse you have before you need to start the treatment. So the reason why most of our devices so far are slow is that we are pumping laterally in the device. We have been pumping and the outlet is out here. We try to separate the loading of the device and the pumping. So if we load laterally, if we bring ions from the side, we fill this chamber, and when we want to stimulate, we just pump out all this vertically. Then we can shrink the dimension considerably in this device. So once again, this is the circuit equivalent model for this. So loading in this direction, the ions comes here, and then we apply a signal, positive signal, and kick out all the neurotransmitter through the outlet. So when we look at the release, this is the recorded amount versus time, and we can see that it's pretty fast. We start to detect even after a few 10 milliseconds. And the synaptic delay in our brain, in the spinal cord, is typically on the level of 2 to 5 milliseconds. We have reached today down to 20 to 40 milliseconds with this. We are within the range where we can have a strong and fast therapeutic effect for epilepsy. However, we are not really competing with synaptic delay. But I would guess we have come down from a second down to this level during 2 years. I'm confident that we actually will beat the synaptic delay or the synaptic speed within a year or two. So to communicate in between these devices, we rely on the possibility that we can have displacement currents in our body fluids. So together with Ericsson, we have been working for almost 5 years to use the body area network technology, where we capacitively send a signal back and forth between, for instance, an iPhone, through the body and to printed electronics or to other devices that are located and positioned on or within our body. So we have produced various kind of labels, including the silicon chip for the communication protocol for capacitive coupling, the antennas to capacitively send signals in and out from our body, and then try to connect this with different kind of devices. This was recently reported a few weeks ago, a few months ago. So now we have a technology where we can ship signals back and forth and to record and actuate functions. Now we have started the work to actually put all this together. So I will switch gear now completely. Plants, it's biological species that lack nervous systems. In order to regulate everything, they rely on basically sending signals in the vascular system, and they also rely on diffusion of these signals. Therefore, plants respond very slowly. However, they have a much stronger response activation scheme as compared to mammalians, or particularly compared to humans. And one reason is that when they are threatened or they're exposed to a pathogen, they cannot run away, they cannot fight back, they have to stand there and rely on a very strong pathogenic response protocol. So typically in these plants, phytohormones, these are the plant hormones, they regulate different processes in the body, and they are sent in the phloem and xylem vascular systems. So these are the communication pathways in the plants that you can use. And they, of course, then regulates via gene expression, growth, physiology, defense mechanism, and so on. So we are aiming at adding on an artificial nervous system, more or less looking like one that we have used in the Biocom lab project, to plants and to see what we can actually use this for. So if you take a look at the plant, it's a biological system including several key components: leaves, stem, and so on. You have the petals, roots, branches. You have the phloem and xylem vascular system. You have the stoma and root hairs that are basically gateways to the environment. The stoma gateway opens and closes and regulates oxygen and carbon dioxide exchange with the environment. And the root hairs, of course, is the component that performs the uptake of nutrients from the ground or from the soil. In some sense, you can say that all these components are analogous to components, interconnects, wires, and contacts, all the four key parts that you need to bring together in order to make electronic circuits. So the idea here is now to use the chemical signals, the tissue construct, vascular system, and overall shape of plants to manufacture organic bioelectronics inside the plant itself. So in the first experiment, we took basically rosa floribunda cuttings, put them into different kind of solutions of conducting polymers. After trying out some 40 different solutions and also killing equally number of plants, we finally found a system, P.SH. This is a conjugated polyelectrolyte. It's water soluble, which did not have a negative effect on the plant. The plant did not die. It performed an uptake, and we could see that we formed very dense, very nice conductors inside some of the xylem vascular channels of the plant. We also performed recording of the conductivity by sticking in probes into the xylem channels. They are typically on the order of 10 to 50 micrometer in width. We recorded then initially a conductivity of 0.1 Siemens per centimeter, not super high for P.dot systems, but high enough to perform devices and other interesting experiments. So in the first test, we then applied, next to the two different contacts, we also applied a third contact in the neighboring xylem vascular system. In this case, the cell wall here defined basically the insulator between the gate and the channel to form a transistor. And we could record very beautiful electrochemical transistor IV curves. And we also recorded fairly nice transfer curves. And we could also see that we have a huge amplification between the gate current, this one, and the drain source current running along the xylem vascular system. By placing two of these transistors in series, we basically define the NOR gate along the stem of the. So this illustrates that we actually can do digital, we can manufacture digital circuits in the xylem vascular system of plants. In the next step, we utilize these stone gateways. Once again, they are the one that open and close to regulate normally gas exchange between the leaves and the environment. These can also be used by shining light. They open up, and then we can use vacuum infusion to basically bring in all kinds of material into the leaves. By doing that, we then focus on P.dot, PSS, composited with nanofibrillar cellulose. And this system you can actually put together to have freestanding, highly conducting system. So this is before the treatment, this is after treatment. The leaf looks a little bit ugly. The good news is that it did not kill the leaf. It survives. The photosynthesis is still running in the leaf. And when we then applied two contacts, we could see that we have an electrochromic effect, because P.dot is electrochromic. When you change the charge accumulation or oxidation level in the material, you can also change color. So this is, of course, an example of an electrochromic leaf where all the display pixels are defined by the veins in the leaf. So by looking at the electrochromic gradients, we can perform potential gradients, and from that we can then basically derive the circuit model for this system. So separated electrochromic parts and then all connected ionically, and ions are represented by these red lines here. So we also apply this to leaves still attached to the petioles, still attached on the stem. Basically, these leaves survives for at least 2-3 months before they naturally fall off, which was a great surprise to us. And we can also basically achieve this electrochromic switching in the leaves. In summary, I will just go through a little bit what we have done. We have basically taken organic silicon-based electronic labels, ion pumps, body-area network to make an artificial neural network. And in plants, we have tried to basically introduce a nervous system using similar devices. And the idea here is, of course, to basically build up a circuit technology systems inside plants. And of course, you ask, why are we doing this? One specific example that we are running in the lab right now is to target the sucrose-gibberellin balance. We are measuring sucrose and delivering the antagonist to gibberellin Gibberellin is a plant hormone that regulates the development process, but primarily it regulates flowering. So it would be nice to see if we can identify when the plant is supposed to flower. If we know cold weather comes in, we can then release the antagonist, and therefore we can then control or delay flowering a little bit. And today this is primarily done by genetic techniques. So I would like to thank all the participating partners in these programs, and by that I would like to thank you for your attention. Thank you very much.